Hello. Today we're going to talk about different strategies to normalize read counts for genetic information between samples. Before we dive into the content, I wanted to highlight this paper, which contains a lot of the information that's going to be presented in these slides. It's a really great read, and I recommend that you check it out. Link in the description. So let's step back for a moment and look at the high level. In terms of DNA sequencing, which is essentially the technology that RNA sequencing is built upon, we're looking at the computational analysis of data, specifically what you do in order to normalize between samples. So in a typical RNA-seq pipeline, you'll have your original raw read data files that you get from the sequencing machine. You'll align those to a reference genome, annotate it, and then as your input file for the normalization, you'll have raw counts, which is the number of runs and reads associated or annotated with different genes. So you have these, in a sense, relative gene counts for different RNAs. And you want to, between different samples, be able to normalize so that you're able to make comparisons between those samples. And the question is, why would there be variation? Well, there's sort of two types of variation. There's variation within the actual data for one sample, and this can be related to, for example, GC content or the length of the different genes reads. So that's one kind of variation. And the second is between differing samples. And one really important cause for the variation between samples is sequencing depth, or the difference in sequencing depth. So on a high level, the sequencing depth is the average number of times that a nucleotide has been referenced or aligned within the genome. So sequencing depth of 100, about every nucleotide will be aligned on average 100 times. Although, as you can see in this histogram, there's a range for how many times a nucleotide is mapped to within the alignment. This looks like a good alignment distribution because you have a pretty, as you can see, a pretty clear center, a pretty clear idea of the sequencing depth. But depending on the conditions of the sample and simply the number of mRNA molecules within it, there can be different sequencing depths, and this needs to be accounted for, obviously, between samples. And this is one of the causes of experimental variability or technical variability, which takes away from the biological significance and thereby makes it more difficult to reach correct conclusions. This is the goal of normalization, to, in a sense, minimize the technical variation and allow you to focus on objects with biological significance. So here are some scenarios and examples that display and show exactly why this is difficult to work with, why normalization is really important. The first concept has to do with the amount of mRNA in the cell, or the total mRNA in a cell. In a sense, the idea is that highly expressed genes could artificially make it seem like there's lower values for other genes, whereas the lower values may not really be lower values at all. It may be simply that there is more mRNA in the cell, and that there's only certain amounts of upregulation and downregulation. So let's make this a little bit more concrete. If you look at, for example, figure A, you see that in condition A, all three of these have this level of mRNA, this absolute level, and that in condition B, 1 and 2 are the same, but 3 is upregulated. But when you look at the proportional shares, if you were to do a relative alignment, you would think, based on this graph here in D, that conditions 1 and 2 were downregulated and that conditions 3 were upregulated, whereas this is actually not the case. So you need a normalization scheme that is going to be able to account for that between the different samples, because that's between the different samples is often where you can get differing total mRNAs per cells, and is able to thereby understand the right conclusions, because you don't really need to write about genes 1 and 2. Those aren't differentially expressed. 3 is your biologically significant difference. It's often helpful to talk about this in terms of upregulation and downregulation, differentially expressed genes. So these graphs aren't related specifically to any of the topics, but they're just some of the representations of differential expression in terms of upregulation and downregulation. Upregulation meaning that the cell is more likely to express something, and downregulation means it's less likely to express something. And these are, as you can see, changes looking at specific genes. Thinking about upregulation and downregulation is going to be important when we talk about the different assumptions that are made by different normalization protocols. Another issue with this principle is that you can't ever quite be sure about the absolute counts. You can't detect a global change in variation. So as you can see here, conditions A and conditions B, 1 and 2, the entire amount of mRNA is different. The condition B, both of these are upregulated. But when you look at this from a normalized read count, when you look at it from a sequencing perspective, it looks like there is no difference. So you can't see a global shift, even though, as you can see in the truth, the global shift really exists. This is a little bit more difficult to fix with normalization protocols, and often can't be done if you don't have controls. This graph will also help to lay out and introduce some of our assumptions, because they're related to symmetry or the amount of mRNA in a cell. So as you can see, A and C, even though there are different genes that are expressed in different ways, the total amount of mRNA when you count up the different bars is still the same. And knowing this allows you to make an assumption to help with normalization. The others is symmetry, the idea that you have the same amount of either upregulated and downregulated genes. And you can see that C and D both have 2 upregulated genes for each condition. This is the kind of symmetry, same amount of upregulation and downregulation that we'll be looking at. And of course, the opposite examples exist for B and D with different mRNA per cell, and for A and B with asymmetry. So the first assumption, same total mRNA per cell, where you assume that between each sample, the total amount of mRNA that you're sequencing is ultimately the same. This is the method. This is the assumption that methods like total count, RPKM, FPKM, the variation ERPKM, and TPM typically make in order to normalize across. And if you have the same total mRNA, then it's easy to sort of just divide by a scaling factor and compare among the different samples. Number 2 relies on balanced gene expression, and this, in a sense, talks about the same amount of downregulation as upregulation, that most things aren't differentially expressed, and that I can figure out and compare between non-differentially expressed genes, between the samples, and thereby create a scaling factor that allows me to normalize between them. Another important assumption here is that the technical variation for a non-differentially expressed gene is going to be the same as it is for a differentially expressed gene. So as you can see in the diagram to the left, this is a different protocol, but 3 different researchers working with 3 different mice, prepared 3 different samples, and then they need to perform one method and try and get the same results. But it's possible that you'll get different results by happenstance, or there'll be variation within the results. This is also filtering into that kind of technical variation, but you assume that the variation as a result of this and sequencing depth, as mentioned before, that those factors impact non-differentially expressed genes to the same degree as differentially expressed genes. And therefore, your scaling factor that you figure out by understanding the non-differentially expressed genes is the same one that you can use on the differentially expressed genes. And there's a lot of strategies that use this: quantile, upper quartile, median D, seq2, TMM, median ratio. Those are all normalization by distribution, where they try and create similar distributions, or looking at, upper quartile being the third quartile and median to try and match all of those up between the samples for expression levels. With regard to this, median ratio is considered to be a more robust form of TMM and earlier protocols that is able to loosen and relax a little bit that other understanding of balanced gene regulation, that there's the same amount of upregulation and downregulation. So you see in the graph below to the right that, the amount of upregulation and downregulation on either side isn't really quite as equivalent, but median ratio is robust enough to still provide a pretty good normalization scaling factor. Poisson-seq and DEGES, those 2 strategies are considered normalization by testing, and therefore are also a little bit more robust in terms of how much you need the upregulated genes to equal the downregulated genes. There's a little bit more flexibility with those strategies. But generally, all of these rely on the assumption of this balanced gene expression in order to thereby normalize between the samples. And the third involves controls, and it actually relies on the same technical assumption that was made in the last slide. The assumption that the technical variation is the same between non-differentially and differentially applies here in a different way. Here we're thinking about that the technical variation between controls and non-controls is going to be similar. And with the controls, there's an obvious implicit assumption that you have controls within your experiment, which you can make happen if you're designing your own data set, if you're designing your own experiment and you're able to keep a lot of replicates and data sets. But if there are cost barriers, or if you aren't able to have perfect controls, then it's sort of difficult to make this strategy work. But controls, in a sense, allow you to more closely get to that absolute value for mRNA. It's not going to be perfect, but it's a better way to get closer than the other normalization strategies. Here you can look at specific housekeeping genes. You can try and use a conventional normalization with spike-ins, that means an increased emphasis on certain genes that you believe are going to be non-differentially expressed, between the two different experimental or not two, perhaps it's more, but between the different experimental conditions. Or you can use factor analysis of controls, which encompasses a wide variety of methods between RUVG, RUVS, RUVR. Typically, you have an understanding of your system to the degree that you can identify different genes, sets of genes that aren't going to be non-differentially expressed between your different experimental conditions. And now this kind of gets to the big question. What strategy should you utilize when you're looking at your data, when you're looking for your experimental conditions, considering everything? There's no one size fits all. I would think about the assumptions and what you have available to you. If you have controls for the data, then you can utilize those robust normalization systems for your specific protocol and thereby create pretty good between sample comparisons. If you don't have controls readily available, then perhaps normalization by distribution, assumption 2, would be better. Or if you know that all of you have the same total mRNA approximately, then maybe the strategies in assumption 1 are going to work for normalization. I would recommend that you think about it in the context of your experiment. In case you want some more advice, I recommend checking out the paper that I cited at the beginning of this video. I'll put the link in the description. The paper includes a table with summarizing a lot of different literature where people would try and utilize the different strategies and rank them based on their applicability to different situations. You can use this table and see whether those applications mirror your experiment or your goal, and thereby look at those rankings and that logic in order to make your own decision as an informed research scientist. Thank you for listening.